Hi everyone, welcome to Brain Tour Part 1. So now we're shifting to the part of the course where we're going to look at physical brain structures and talk about their function, how things are classified in the brain, and anatomical directions and things like that, leading up to the first practical exam. So this is definitely an important lecture. I would highly recommend that you refer back to it, make sure you really have a deep understanding of it. And if you have any questions, please feel free to come to office hours or send me an email. It's very important that you understand the functions and physical locations of the structures we're going to go over. So without further ado, what are the differences between the sheep and human brain? So sheep brains and human brains are obviously quite different, but they do share some important similarities. But generally speaking, the human brain is larger, and if you can see, all these wrinkles and grooves, sulci and gyri is the other word for them, are very pronounced in the human brain, whereas they're not as pronounced in the sheep brain. And sulci and gyri essentially allow us to have more surface area in our brains. So we're packing in as many neurons as we possibly can and increasing our computational power as much as possible in a finite space. So if we can't expand outward, we have to go inward. And so more grooves to give us more ability to have more neurons to work with. More power, more neurons, more computational power. Another thing that makes the human brain different is that our frontal lobes tend to comprise a much larger part of our brains compared to other animals, and sheep especially. And sheep have a larger olfactory bulb and better sensory regions compared to humans, which would make sense since they work more with their noses and other sensory regions than we do. However, while the volume of certain regions like the olfactory bulb or the frontal lobe may differ, the functions are still the same, which makes them a perfect model for dissection. And this is why if we were taking this as an in-person version, we would be working with sheep brains because they do look and act so similar to our brains. They are they're like a little teeny tiny version of our brains, with some important changes, but functionally speaking, everything is there and everything works the same, even if the volumes are different. But before we move forward in talking about brain structures, we need to lay down some rules. More like need to lay down some directions. So when we're doing dissections or talking about brain structures, if you're reading a journal article or watching a video, like if a surgeon, if you're I've seen some videos of surgery or doctors talking. So if you do that, they may use anatomical directions, which can be kind of confusing because they're not north, east, south, and west. But using these anatomical directions can make things easier to identify, what region, what area, or what action we're completing in the brain or the body. But for dissection specifically, if we say something like, make an incision anterior to the optic chiasm or posterior to the cerebral peduncles, then you'll have a better understanding of where that cut should be and where it will go and where you're going in the brain. So let's talk a little bit about anterior versus posterior, or the rostral caudal. These are basically synonyms of each other. Most of the time in journal articles, which is where I see these directions the most, I see anterior and posterior, but it's good to know both. So anterior or rostral is toward the nose, or rostrum is another word for beak, and posterior or caudal is toward the tail or the bum. So when I was learning this, I heard caudum, I thought caudbum, to think of the tail. So, or posterior, I think of, a very prim and proper British woman saying, "Don't let out your posterior." I was not trying a British accent, but, someone a little stuffy, instead of saying but or any other word, they would say, "Your posterior." So anyway, what about dorsal versus ventral? So we have dorsal, which is toward the back or toward the top of the head. So the dorsum meaning the back, and ventral, which is toward the chest or the bottom of the head. Obviously, our focus here is a little bit more toward the brain. So I think of dorsal, top of the head, ventral, going toward the chin or the bottom of the head. I really like this image of I'm going to say it's a lizard that shows anterior, posterior toward the tail, ventral toward the tummy, and dorsal toward the top or toward the back. And breaking this down even further, we have another set of anatomical directions to think about: medial and lateral, and superior and inferior. And definitely we'll be hearing these a little bit later in this lecture. And for medial and lateral, if you visualize for a moment that we have this little line through the center of our bodies, then that would be medial, toward the midline of the body. And our imaginary or not so imaginary line is the spine. So medial structures are going toward the midline or our spine. And they are closer to the medial longitudinal fissure, which is the main fissure that separates the left and right hemispheres of the brain. Lateral is the opposite of medial, meaning that it's away from the midline of the body or moving toward the sides. So, medial is your spine. Moving laterally, you're going toward your fingertips because they can reach outward far, far away from the spine. So for example, we have the medial temporal lobe. That's the part of the temporal lobe that's very deeply inside the brain. It's not a surface structure because it's medial, and it's closer to the midline and spine in our bodies, and it's a structure that is responsible for certain types of episodic and spatial memory. And the superior versus inferior, this is for humans and primates since we do walk on 2 feet. For the superior direction, that's toward the top of the brain, so above things, or you're inferior to something, or you're toward the bottom of the brain, or you're below another structure. 2 structures that will use this nomenclature are the superior and inferior colliculi. The superior colliculus is superior to the inferior colliculus, meaning it is above the inferior colliculus. The inferior colliculus is below the superior. And that sounds confusing now. Just kind of keep it in your mind. We'll come back to it a little bit later and you'll see what I mean. So now just to round it off, I really, really like this image. I'm almost certain I've seen this in one of my own courses. And I like how it brings everything in from rostral or anterior to caudal and posterior, dorsal and ventral, from the midline, either from the brainstem or from if you're thinking more anterior or posteriorly, or and then superior and inferior. I think this is a really great way to visualize what we're going to be doing in the brain and talking about. So keep all of these, refer back to them. Over time, you'll get really comfortable with it and it'll come naturally. But it takes a moment. It takes some time. It is kind of confusing. So give yourself the time to get used to these directions, and then after you know it, it'll be all normal. So now we need to cover planes of section, how things are cut or dissected for the brain. And we have several different cuts here: the horizontal, coronal, sagittal, mid-sagittal, and cross-section. So a horizontal cut is actually not a cut that we'll look at this semester, but it's still important to talk about. And it's cut in a plane that's parallel to the top of the brain. And cross sections are actually a type of horizontal cut that are done on long or in narrow structures. And then we have the coronal section, which start very laterally and then go in more medially, and they are cut in a way that's parallel to the face. So basically the opposite of a horizontal cut, which is going sections from the top of the brain. This is going sections from, if you were going from your face and then making sections, that is how it's going instead of horizontally. And for the sagittal cut, we're then cutting in a plane that is parallel to your ears, and which begins anteriorly, and then becomes more posterior as you cut. And a special cut that we're looking at in this course is the mid-sagittal cut, which is that cuts straight through the middle or medial longitudinal fissure, which again is that main line that separates the right and left hemispheres. So basically cutting the brain in half. And as I said a little bit earlier, cross sections are the type of horizontal cut that are done at a right angle or 90 degrees to any long or narrow structure. So basically the brainstem and spinal cord is where you would see these cross sections. And to kind of make this a little bit easier to visualize, here using mirrors to show where a sagittal cut, horizontal, coronal, and cross section cut would occur. And this is also then done, this is actually on the right, the left is a rat brain, showing where this rostral to caudal, the mid-sagittal cut, horizontal, and then coronal cut. And now we're going to start with our first section, or our first layer of the brain, a surface structure. And when we think about the brain, we're not normally thinking about how, we think of it just as like this jelly that's sitting in our skulls and that's it. But actually we have the meninges, which are 3 outer membranes that protect the brain from basically bumps and scrapes. Each individual layer will look different and will protect them in a different way. So first we have the dura mater, which is the outermost protective layer. It's really tough and leathery. It's kind of translucent, not completely opaque. It restricts the movement of the brain in the skull. So when you bump your head, this will help your brain stay in place and will also prevent blood vessels from breaking if you have a really severe fall or some sort of head trauma. And then we have the arachnoid mater. Oh, and to mention, the dura mater is the only meninges layer that you'll physically have to identify in a practical exam, but you should still know the function, how the other layers generally look. So anyway, our second layer is the arachnoid mater, which is spongy and kind of spiderweb-like. It's thinner than the dura mater, not as leathery. It is quite spongy when you touch it, and it really does look like kind of like veins like in a spider web. And it's white. And this will essentially act as a cushion for the brain during harsh contact. So when your brain takes a tumble, the arachnoid mater will try to soak up as much of the impact as it can, so then your brain is hitting something that is soft rather than your brain, or even if you got hit hard enough, the dura mater, which would be very tough and inflexible. So even though that we talk about 3 main layers, there is a middle layer between the arachnoid layer and the pia mater, which is called the subarachnoid space. And this contains cerebral spinal fluid and arteries, and the cerebral spinal fluid flows through the circulatory system or through a circulating system or ventricles and helps create buoyancy. And there are also holes in the or it creates holes in the pia mater which can allow the cerebral spinal fluid to get nutrients. So then finally, we have the pia mater, which is the thinnest, most delicate layer of the meninges. Kind of looks like laminate or plastic cover. And it keeps the cerebral spinal fluid in the correct places rather than just flowing every which way around the brain and just going everywhere instead of being concentrated where it needs to be. And can be pierced by blood vessels in the subarachnoid space, which will allow cerebral spinal fluid to get nutrients. And as a final note for this, you might have heard of the illness meningitis, which is a swelling of the meninges. And this is so dangerous because the meninges they're basically right on top of the central nervous system. So when these are infected, that can put a lot of undue pressure on the brain and can cause damage, and you can be very, very sick getting meningitis. And here is a sheep brain. And this is the dura mater. As you can see, it's translucent, kind of leathery. If you could touch it, trying to give the best sensory description that I can. But this is the only structure of the meninges that you would need to identify. So now we're going to talk a little bit more about the major divisions about the brain. So we have three main divisions: the forebrain, midbrain, and hindbrain. But then we break them down further into the telencephalon, diencephalon, mesencephalon, metencephalon, and the myelencephalon. It will definitely be important to memorize these classifications. So where is the telencephalon, myelencephalon, and what structures are within these classifications. So the first division that we're going to talk about is the forebrain, and within that is the telencephalon, which is the cerebral cortex and hemispheres, technically. A good way to remember this is thinking tel and telencephalon, tel as in top. And as you can see, there are tons of folds and wrinkles, which is typically what we associate with an image of a brain. And these are the gyri and sulci. So a gyrus is a protuberance on the surface of the brain, and the sulci are the folds and grooves. So technically, this is obviously not a perfect rendering, but something like this right here would be a gyrus, and then like down here, it's kind of U-shaped curve would be a sulci or sulcus. And we have the cerebral cortex within the telencephalon, most importantly, which is separated by two hemispheres to the left and right, which medial longitudinal fissure will divide the cortex into two hemispheres. And then we have the lobes after that. And there are 4 lobes in total, and we'll talk about them in just a moment. No worries there. And when we talk about the cortex, we're thinking about this outer gray matter, the wrinkly part. But when we're talking about the lobes, we're talking about gray matter, wrinkly stuff, plus everything that's on the inside more medially and on the outside. And then in addition to the cerebral cortex, we also have the olfactory bulb and tract in the telencephalon, which receives input. So the bulb, receives input from olfactory or smell receptors in the nasal mucosa, and the tract has axons that relay input from the olfactory bulb to areas in the cerebral cortex. So it goes olfactory bulb to olfactory tract, which then will go to the olfactory cortex in the frontal lobe. This is just an image of the medial longitudinal fissure and how you would see it on the exam. So if there were a bunch of pins down the main line of the brain, this is the medial longitudinal fissure, which the function of it separates the left and right hemispheres of the brain. And these are also the olfactory bulbs. So, well, technically, and this is kind of a problem of having images to rely on instead of having a dissection. So you could look at this, and mess with it with your hands. This is technically the olfactory nerve, which we'll talk about next week. And the bulb is right under it. And it's just something that we have to deal with. But if you saw something like this on an exam, and it wasn't specifically looking for a nerve, this would be the olfactory bulb. But I'm hoping to get something a little bit better to use. Anyway, now we can talk a little bit more about the diencephalon, which is comprised of the thalamus and hypothalamus. So the thalamus is the relay center for the brain, which sends necessary information to the right areas for processing. So essentially what happens, so the thalamus will receive some sensory information, maybe about touch, and then send it to the correct area, so the somatosensory cortex for touch. And then we have the hypothalamus, which is below the thalamus. It controls the autonomic nervous system and organizes behaviors that are related to survival. You may have heard this little phrase associated with the hypothalamus called the 4 F's, that are the main actions that the hypothalamus is responsible for. So fighting, fleeing, feeding, and fornicating. So it's, hypothalamus is, things that we don't necessarily think about and control, but things that we need to survive and keep going on day after day. And the hypothalamus regulates these 4 F's by controlling the release of hormones by the pituitary gland. And these will help us to maintain homeostasis. And then we also have the optic chiasm, which is a region where optic fibers cross over to the opposite side of the brain and contains nasal retinal fibers. I have a picture of this later in the lecture, so don't worry, that sounds confusing. I promise it's not as confusing as it sounds. You just need something to visualize it. So hold that thought for a few slides down the line. So nerves or fibers, will stay on the same side, so ipsilaterally. And then this is the reason that the right eye can be processed by the left hemisphere and vice versa. So there are portions of it. So nasal retina is ipsilateral and it is not ipsilateral. The nasal retina is contralateral, and then outside toward the temples is ipsilateral, and nasal retina is contralateral, meaning that they do the crossing. Don't worry, that sounded even more confusing. I apologize. I promise very soon I will show you exactly what I mean. So moving on, this is an image of the pituitary gland and pinned. So you can see that some of the dura mater is still attached, and this is because when you remove the dura mater, the pituitary gland is also removed. So in order to pin it, we have to keep just enough that we can take a picture of it so you can see. It's just a little button right by the optic chiasm. And speaking of the optic chiasm, here we are, pinned image and the image I told you that we would see in just a moment. So the optic chiasm, this image isn't perfect. You can kind of see that little X shape here. That is the optic chiasm, and in the image, it's right here where the nasal and temporal retinas are, where they cross over. So you can see the nasal retina closer to your nose. They cross over, while the ones closer to your retina stay ipsilateral or on the same side. So nasal contralateral, temporal, ipsilateral. Not a big deal to know, but when I first learned this, I also learned that at the same time. So by process of habit, I wanted to tell you all about that too. But, the important part of this is if you saw a pin in the kind of like an X-shaped structure, then we would have this would be the optic chiasm. So moving on a little bit, we have the lobes. So this is kind of changing a little bit from talking about the major classifications of the brain. This is something you'll hear pretty commonly, so I would highly recommend memorizing this. So we have 4 main lobes to talk about, and technically 3 different sulci to look at. So we have the frontal lobe, parietal lobe, temporal lobe, and occipital lobe, and we'll go over all of the functions of these in just a moment. The main sulcus or division that you need to really know is the central sulcus, which is right here, and the important part is that it's adjacent or posterior, technically, to the primary motor cortex, which is necessary for executing motor movements. So the primary motor cortex should be right here in a band. And then the lateral sulcus, I don't know why this image has it here. This arrow should be more here, between the parietal and temporal lobes. But the lateral sulcus will divide the temporal, parietal, and occipital lobes. And then finally, back here by the occipital lobe, we have the occipital sulcus, which separates the occipital lobe from the parietal and temporal lobes. So this is definitely another important image, but broadly speaking, if you take another neuroscience or brain-related course, these are all important to keep in mind when you're learning new information about the brain. So let's talk a little bit about the lobes. What about the frontal lobe? The frontal lobe is very popular because it is kind of what separates us from all the other animals in the animal kingdom. It's involved in motor processes and executive functioning or higher order thought, which just means that the things that make us more complex than other animals, I guess, from a thought perspective, happen in this region. So there are 2 important anatomical classifications. So the anterior portion of the frontal lobe and the posterior portion are responsible for different things. So remember, the anterior portion is closer to your nose, and the posterior is closer to your behind or toward your back. So looking anteriorly, the anterior prefrontal lobe contains the prefrontal cortex, which is our magical executive function area, which separates us from all the other animals, which it's involved in planning, decision-making, consequence learning, complex language, emotional inhibition, and other functions that are considered higher order learning. So that's what you can use. And then for the posterior frontal lobe, that contains the aforementioned motor cortex near the central sulcus. So if you're thinking in terms of the motor cortex is anterior to the central sulcus. But if I said, oh, well, where is the central sulcus in relation to the motor cortex? You would say, well, the central sulcus is posterior to the motor cortex. Like I said, it's a little confusing, but once you get used to it, and once you get used to it, it'll all be fine. And most animals have a much smaller prefrontal cortex volume than humans do. We are just jam-packed with prefrontal cortex. It is a huge portion of the frontal lobe and arguably a very important one as well. So, also, something else to note. So impulsivity is a big deal. There is a strong relationship between another structure we're going to talk about soon. So the prefrontal cortex and the amygdala share an interesting relationship where the prefrontal cortex inhibits the amygdala. So for fear learning and inhibition, so the prefrontal cortex inhibits the amygdala in a way that makes us and other animals less impulsive. So, the long story, make a long story short, it's to stop animals from making stupid choices, either based on fear or not based on any fear. So when you have a damaged prefrontal cortex and your amygdala is just running wild, a way that you can see that in your behavior is strong impulsivity. That's just a interesting aside. Anyway, here is an image of the frontal lobe. So it'll be very anteriorly pinned. So if you see something that looks like this, I kind of think it looks like two little eyes connected to with how it looks pinned in the angle of the image. But don't assume that it's going to look just like this, but the pinning should anyway. But if you see something like this, definitely frontal lobe. So now we've talked a little bit about the frontal lobe. What about the parietal lobe? And as you can remember, the parietal lobe is posterior to the frontal lobe. And technically, it is dorsal to the temporal lobe. All right. So the parietal lobe is important for somatic sensations and spatial orientations, and that's because in the anterior portion of the parietal lobe, we have the primary somatosensory cortex, which is directly posterior to the central sulcus. So we have motor cortex, central sulcus, somatosensory cortex. So here we take in information about touch, texture, temperature, and pain information when we touch things. And the parietal lobe is located in the dorsal posterior portion of the brain and also contains the primary somatosensory cortex. And the parietal lobe monitors all information about the eye, the head, and body position and will pass it on to areas that control movement. So perhaps some of this information will go to the primary motor cortex near the central sulcus. It is also essential for spatial information, touch from the somatosensory, and importantly, numerical information, so processing numbers, attention, vision, so object locations relative to yourself. So the parietal lobe is extremely important for daily function. And here is an image of the parietal lobe and how you would see it. So if you have pins or dots here, then you're definitely looking at the parietal lobe. So up next is the temporal lobe, which is important for learning, memory, and hearing. Sorry, I just made this a little bit easier to read. So this is a quite ventral structure. It is beneath both the lateral and central sulcus and the parietal lobe. And this is the structure that extends the most laterally in the brain. And it does an absolute metric ton of stuff, especially within the cognitive and behavioral neuroscience program and the IPN, or the interdisciplinary program. We love the temporal lobe, especially because it is such an important area or region for learning and memory. And it contains the primary auditory cortex for processing auditory information in addition to language. And it's important for face recognition, so the fusiform gyrus is contained here. And if you damage the fusiform gyrus, you can actually get prosopagnosia, which, don't quote me on that pronunciation, is a little shaky. But this disease essentially is when you can't recognize someone's face. And one of the most famous cases of this was detailed in Oliver Sacks' book, The Man Who Mistook His Wife for a Hat. And the title, while it sounds funny, is actually quite literal, that I believe it was the left visual field. So if his wife was standing in the left visual field, he would mistake her for a hat or an object that was nearby. You're like, that's not my wife. That's a literal hat. It's a fascinating book to read. I highly recommend Oliver Sacks' stories if you're interested in seeing some of clinical cases of how the different types of brain damage or brain abnormalities can present. Very interesting stuff. So additionally, damage, we also have the medial temporal lobe to back it up a little bit, or the MTL, which contains the entorhinal cortex, hippocampus, and amygdala. And these are very important for learning and memory, so spatial, episodic, and temporal or time memory. So if you damage or remove the medial temporal lobe, it will lead to deficits in the aforementioned spatial and episodic memory. A famous case that you will either learn about, have learned about, or will learn about is actually HM. He had most of his entorhinal cortex and medial temporal lobe removed, and it caused broad deficits in his ability to form new memories. So he basically, this happened because he had very bad seizures when he was 16. So he had surgery done, and he couldn't remember anything from before being 16 years old. So forming new memories was just not possible. A fascinating case of how interconnected our brains are, and that memory is not just something with the hippocampus. The entorhinal cortex is really important. The neocortex is important. This is kind of an aside, but everything is interconnected and nothing happens in isolation in the brain. Anyway, this is a pinning of the temporal lobe. So how I think of the temporal lobe, or how I remember where it is, I think auditory cortex between the ears, there's the temporal lobe, and that's how I would remember this pinning. And now we have the occipital lobe, all important all the way in the back, and highly important for vision. So it is the most posterior portion of the brain and contains the primary visual cortex, which receives input for complex aspects of sight. And destruction of any part of the striate cortex, or the primary visual cortex, which is another name for it, causes cortical blindness in the related part of the visual field. So moving on, if you see a pin here, this is kind of confusing for some, but if you see a pin here, this is the occipital lobe. This structure right below it is the cerebellum. Completely different structure, interesting one, it's great to learn about. However, if we have pins up here, it's for the occipital lobe and not the cerebellum. If I were to see an answer like pins were here, and you're like, that's the cerebellum, that's not the cerebellum. Another name for cerebellum is little brain. So downtown is the cerebellum. And now we're going to move to the next classification, which is the midbrain, which contains the mesencephalon, which has the tectum and tegmentum. They are very similar in name, but don't get them confused. So the tectum is a dorsal part of this, so the roof, which contains the superior and inferior colliculi. So the superior colliculus processes simple aspects of visual stimuli, and this inferior colliculus does the same, so simple aspects of auditory stimuli. So the superior colliculus will go to the occipital lobe, we'll send information there, to the primary visual cortex, and then the inferior colliculus will send information to the auditory cortex for more complex processing. The superior colliculus will be active if something like is in, you're a part of your visual field that surprises you. It'll activate really quickly, and then you'll turn toward it to make sure it's not dangerous. Same thing with your inferior colliculus. If there was a startling sound outside of your field of view, then you would hear it, your inferior colliculus would activate, you'd turn toward it, you're like, oh, is that a dangerous sound? Should I be afraid? And then, so that's part of the reason why that action happens. So further, we have the tegmentum, which is the ventral part of the mesencephalon, and which contains the cerebral peduncles. And these are motor system fibers from the cerebral cortex that project to the brainstem and spinal cord. So here is a pinning of the superior and inferior colliculi technically. And with the yellow pins, we have the superior colliculus, and the black pins, which are a little hard to see, are the inferior colliculus. So with this image, it's a little tough to see, but when I was learning this, I heard that the superior and inferior colliculus are like a butt and a toilet seat, so that the superior colliculus is the butt and the inferior colliculus is the toilet seat. And that image was so ridiculous to me that I have never forgotten it all of these years later. So that might be useful for you. I wish I had a little bit of a better image. So I just paused for a moment. This is another image that I had found online of the superior and inferior colliculus. This is a drawing, of course, but this kind of shows that image that I was describing a little bit ago. So that might help make this a little bit easier to remember. But stuff like that is helpful for my learning. So if it's helpful for you, I am more than happy to share. Sorry it was a little ridiculous, but... So anyway, let's move on. Here is a pinning of the cerebral peduncles, and this is close to where the pituitary gland was, so the dura mater is now removed. So you can see right in the center between these peduncles would have been where the pituitary gland was, near the optic chiasm. So once the dura mater is gone, you can see the peduncles. And now we can move on to the hindbrain, which is the metencephalon and the myelencephalon, which contain the cerebellum and the pons for the metencephalon, and for the myelencephalon we have the medulla. So the cerebellum is crucial for coordinated motor functioning, so movements that require precision, and will also coordinate with the vestibular system to balance these precise movements. There was actually an interesting case study where someone was born without a cerebellum and had a pretty normal life. They didn't even know that they were missing a cerebellum until they had a brain scan in adulthood. And the researcher was kind of freaked out because they were missing a whole brain structure, and that was not expected. But the only real difference was that they were kind of clumsy, and this can be attributed to the brain being able to account for it since this structure was gone since birth. So some neurons and pathways might have accounted for it early on in life. That made this easier to deal with. If someone brought you into surgery right now and said bye-bye to your cerebellum, you would not, just kind of be a little woozy or clumsy. This would be very, very rough for coordinating your movements and having some sort of balance. So it's definitely a necessary structure, is my point. There are also some case studies in research that says the cerebellum also may be further involved with cognition, but we know for certain that the cerebellum is very important for coordinating motor function. So for the pons, that relays sensory information between the cerebellum and the cerebrum, meaning the rest of the brain, and is involved in controlling your breathing, and is ventral to the cerebellum. So what about the myelencephalon? This has the medulla, which controls autonomic functions like breathing and blood pressure. It's a long, narrow structure. So what type of cut would we have here? If you guessed a cross section, you would be correct, which is a type of horizontal cut. So it has a lot of white matter carrying information to and from other brain areas, and the pons and medulla together are called the brainstem. This is very, very important. Actually, I'm going to make the word for this week medulla. So our word for attendance this week will be medulla. Alrighty, moving on. This is a pinning of the cerebellum. So pin here, cerebellum, pin up north a little bit. That's the occipital lobe. Here is a pinning of the pons and medulla respectively. So the pons is kind of like a little bump basically right next to the medulla. So they are like two peas in a pod, and together they are the brainstem. So finally, here we are coming to the end. This is for the lab practical. So make sure to point out all of the physical locations. So anything that we talked about during the lecture for this week could be on the practical exam, but definitely make sure you know all the physical locations on this list. All of them might be, probably will be on the exam. Hint, hint. So make sure to know them, please. Also make sure to know something about their function and what they're responsible for. So where they are in the brain, a little bit about what they do, and then we're all good. So just to remind you, the word for this week is medulla, so MEDULLA, and I will be posting the lab report for this week. Honestly, doing the lab report while watching this is probably a good idea, and I'll put that in the directions. And if you have any questions about the structures or their function or anything about that, please send me an email, drop by office hours. I'm more than happy to help out. I want to, smooth out any kinks or any confusion here. It's important to know all these structures and what they do. Anyway, thank you so much for listening, and I will see you next week. Take care, everyone. Bye.